Good evening, my name is Mariana Nava, and I'm going to present you about Maguey, Agave salmiana, micropropagation. So, Agave salmiana, most commonly known as maguey, is a Mexican endemic plant, which is mainly produced in the states of Hidalgo, Tlaxcala, Estado de Mexico, and Puebla. It has a medium to large size, and it has been used for textile fiber production, pulp elaboration, as well as gastronomical and ornamental uses. It is a Mexican endemic plant that has the ability to adapt to stressful environmental conditions. It produces high-quality meat, and all its parts can be used, even its pests. By micropropagating these species, we will be able to obtain populations with elite characteristics, as well as to obtain plants with antioxidant activity. It is also important to note the key role that maguey plays in the Sustainable Development Goal number 15, which is life on land. Maguey crops protect the land against erosion, helping it to conserve soil's nutrients. And it also captures and restrains moisture that is used by adjacent crops. It is also host to animals and insects, such as pollinators. Maguey has been shown to have cytotoxic activity from extracts obtained by agave sap and leaves. We can obtain fructans and glycosides that have been shown to have a cytotoxic activity in these particular cell lines. The objective of this project is to design and implement a methodology for the in vitro micropropagation of Agave salmiana in order to obtain good quality plants, to evaluate its potential cytotoxic activity to treat HEC293 cells, ensuring that the findings will be beneficial to our training partner, WeClone. WeClone is the first commercial micropropagation laboratory in Mexico. It is located here in Querétaro, and it was co-founded by Dr. Ashutosh Sharma. Our hypothesis is that the treatment with a specific combination of hormones, IAA and BIP, will positively influence shoot induction and efficient multiplication of agave salmiana plants in a micropropagation system. For our methodology, we had several methodologies because we had a lot of issues with the cleaning process. So we used different media preparations. On the first preparation, we only use MS, the hormones at its particular concentrations, agar, and we adjusted the pH to 5.7. For the second preparation, we decided to add activated coracle to combat oxidation and to add two different antifungicals, Captain and Benlate. However, by adding these antifungicals, we lowered the pH of the media, which meant that it did not solidify. So for the third preparation, we decided to remove these antifungicals. For the fourth methodology, that it's our final proposal, we will be using the third media preparation for the first cultivation of the explant, and we will then subcultivate it to the first media preparation. So we had different cleaning processes. For the first methodology, we had shorter times for the washes, and we also had like a really general cleaning process. So for the next cleaning process that was done for the third methodology, we decided to add a wash with antifungicals, Captain and Benlate, as well as a wash with microdin. It is important to note that we did not have a cleaning process for the second methodology due to the fact that the media did not solidify, which meant that we were not able to clean or cultivate any samples of agave salmiana. So for the fourth and final methodology, besides the bath of antifungicals and the microdin bath, we also decided to add an antioxidant bath. This is the final methodology that we proposed. First of all, is the mother plant selection that it was done by our training partner. We then remove the leaves and the roots of the plants. We do first a superficial wash with twin 20, and then a 3-minute wash with twin 20 at 2%. We then perform the extraction of the mesosomatic explants, and we have first a wash of 1 hour with antifungical, Benlate at 2% and Captain at 2%. And we then have a 1 hour wash with microdin at 2%. After this, we have a wash of 35 minutes with 40% chlorine. And finally, an antioxidant bath. This was done with citric acid and absorbic acid at a concentration of 150 milligrams per liter. We finally removed the damaged tissue and we put the explant into the media. For the cytotoxicity assay, we will be using A0293 cells, which are human embryonic kidney cells. For the extract preparation, we first wash the leaves. We dry them at 40 degrees Celsius for 8 days. We ground them and we mix 10 milliliters of methanol with 1 gram of powder. We place it on the sonicator and we centrifuge it. And we finally filter it in order to sterilize it, and we store it at 4 degrees Celsius. For the cytotoxicity assay, we'll be using 4 different concentrations, as well as a positive control and a negative control. And we will be reading the absorbance at 517 nanometers after 24 hours and after 48 hours. For the first methodology, we had a lot of contamination. We had bacteria and fungi, and we also had oxidation in the explants. For the second methodology, as I have mentioned previously, the media did not solidify. This was due to the fact that by adding the fungicides, we lowered the pH from 5.7 to 2.8, 2.1 after the autoclaving process. For the third methodology, we had 1 healthy explant, 1 positive result, but we also had fungi contaminations and oxidation. For the fourth and final methodology, by adding the antioxidant bath, we were able to obtain positive results. However, these explants were only healthy for about 2 days. So we had a subculture. We first cultured the explants in a media with activated charcoal, and we then subcultured them to a media without activated charcoal. It was done after 4 days in the case of all of the explants of the fourth methodology, and it was done after 13 days in the only surviving explants from the third methodology. So after 6 days of this second subculture, we can see that all of the results obtained from the fourth methodology resulted in necrosis, but the positive result we had from the third methodology did survive. So we can see that mainly in the early stages of the project, we had a lot of bacteria present, and this was present in all of the explants, which leads us to believe that it was due to endogenous bacteria, which in the case of Agave salmiana is Bacillus, Enterobacter, and La Klebsiella. We also had fungi, but this was only present on a handful of explants, which leads us to believe that it was due to poor aseptic technique. Oxidation is a frequent problem in vitro culture, and it is due to the fact of the presence of damaged tissues. These damaged tissues can be obtained from aggressive disinfection processes or from the cuts made to the explant. And this results in the production of phenolic compounds, which will be absorbed by the media and will lead to inhibit growth and to tissue's death. For the cytotoxicity assay, we can see that all of the percentages of viability obtained after 24 hours and after 48 hours were always above 70%, which means that the concentrations used were not cytotoxic. If we want to further research the cytotoxicity activity of these plants, we will need to use different extract preparation, as well as different concentrations and more time of exposure. For the statistical analysis, we first wanted to test the difference between the mean of the number of shoot formation per explant in the different treatments, but it was not possible to do so due to the fact that no growth was obtained because of the complications of the cleaning process. But ideally, if we had obtained this data, our response variable will have been the number of shoots grown per treatment, and we will have done an ANOVA of 1 factor for levels. We also wanted to test the difference between the mean of the absorbance value read at 517 nanometers for the different concentrations for the cytotoxicity assay. But it was also not possible to do so due to the fact that we did not have enough repetitions. So to conclude, we were unable to test the effectiveness of the treatments due to contamination and oxidation problems, but we were able to develop a cleaning method that successfully addressed the bacterial and fungal contamination. However, we will need to do further research in order to combat the oxidation process. We also need to expand the test with different concentrations of the extract or using a different plant source to test the cytotoxicity of agave salmiana. And we will also need to do more replicates and to continue researching in order to obtain significant data to have a statistical analysis. That will be all. Thank you very much for your attention.